short talks and then a final lecture before closing remarks. So without further indulge, let's get started. Our first speaker is Carl Philipp Heisenberg from ISTA, Austria, and we'll talk about establishing oocyte cell polarity.
Okay, thanks very much for the invitation. The title I have not chosen. I got this title, I think. So what I'm really talking about is ooplasmic reorganization and cleavages in zebrafish, and not the other title. I'm speaking about very shortly about published work from a former PhD student in the lab, Cheyenne. He has left in the meantime and is in Lukas Peckmann's lab in Zurich now. And I will mainly talk about unpublished work in progress by a current postdoc in the lab, Nikhil Mishra. What I would like to do is speaking first about how the oocyte, how the egg, the laid egg, segregates its yolk granules and ooplasm. And what you have is, this is a zebrafish egg. It's just being fertilized, and it has essentially two components. One component is a yolk-free cytoplasm, which has already a bit accumulated up here at the animal pool, and then in blue more you have the yolk granules. And what is happening then, and this is a process which has been described for many years already in zebrafish, it's called ooplasmic streaming. What you see is that the ooplasm streams up, accumulates at the animal pool where the first cleavages is happening, and the yolk granules are accumulating on the vegetable pool. So you get a segregation of two phases. One is yolk granules and the other one is ooplasm. And this process is somehow accompanied to the onset of cleavages and embryonic development. Now, as I said, this is a process which had been looked at for many years, and the prevalent idea in the field when we started to look at it was that this ooplasmic streaming is achieved by a process like bleb formation in migrating cells. Essentially, what has been proposed is that you start with an egg, a laid egg, which has a uniform actomizing cortex, and then what you do is, for initiating ooplasmic streaming, is you reduce actomizing contractility, the actomizing cortex at the animal pool selectively. This leads to a deformation of this actin cortex because it becomes weak, and then preferentially ooplasm is flowing in and leaves yolk granules behind. So that was sort of the idea, and in principle this is not completely wrong, because when you look at the actomizing cortex, there's indeed a little bit of a weakening coinciding with the deformation here. So when Cheyenne then joined my lab, he's a physicist from training. He wanted to put these eggs into square boxes and wanted to spatially confine them and see how flows are being regulated in spatial confinement. He didn't do so well, and what happened then sometimes is that these eggs were exploding and you ended up as a soup of yolk granules and ooplasm. That's not what he really wanted to look at, but what he found to his surprise is if you destroy these eggs and you don't have a plasma membrane, you don't have a cortex, you certainly don't have blebs, the ooplasm is able to self-organize in a way which looks quite similar to what the fertilized egg is doing. You get streams of ooplasm up and you leave the yolk granules behind. And that sort of indicated that this segregation process is really a self-organizing property of the ooplasm and yolk granules, and it doesn't need a plasma membrane, it doesn't need a cortex to occur. Now the question then he asked, of course, is how is this self-organizing property regulated? And what he noted is, if this is again a soup here, and what he labeled is actin in green, and what he found is that at the former animal pool of this exploded egg here, there is a bulk actin polymerization wave originating and then traveling into this soup of yolk granules and ooplasm. That's sort of interesting. It's something which is not only occurring in these yolk granule ooplasm soups, but it's also something happening in the intact egg. If you look here at the intact egg, you see this bulk actin polymerization wave originating from the animal pool and then traveling into this fertilized egg. So this wave looked interesting. The question is, how would this wave, or would this wave at all be related to the streaming process which is occurring at the same time? And what he noted then very quickly is that this wave actually leads to a preferential accumulation of actin at the animal pool, where in general not only bulk actin but also cortical actin is higher, and this leads to a gradient of actin along the animal-vegetal axis with high actin at the animal pole and low actin at the vegetal pole. And if you have a gradient of actin, and even if myosin 2 is uniformly distributed, you still get flows. And that's something he noted, that there is actually actin flowing, and this is an actin label, and it flows from the vegetal pole up to the region where there's more actin to start with, where the gradient has its peak. So what he noted is that there is, and this is another actin movie here, that you get considerable actin flows, bulk actin network flows, originating from the vegetable pole and going up to the animal pole. Now, the question then asked, and I will be very short here and have just a summary slide, how would this flow translate into segregation of ooplasm and yolk granules? And what we postulated, and this goes back to a long-standing collaboration with Eduardo Hanesso, who is a physicist, a theoretical physicist at ISTA, where we speculated initially and then showed an experiment that this is indeed, that we have supporting evidence supporting these ideas that there is differential friction. And the idea is that once you have an actin network flowing in a specific direction, it can take along either the yolk granules or the ooplasm. But the interface to the ooplasm is much, much larger than the interface to yolk granules, because yolk granules are very large objects. So you have a very large interface to ooplasm and a smaller interface to yolk granules. This would translate into something which you might call then differential friction, where the actomyosin network takes preferentially along the ooplasm and leaves behind the yolk granules. And that, in a number of experiments I'm not showing you, seem to be the case that this segregation is really driven by these actomyosin flows, and these actomyosin flows take along the ooplasm and leave behind the yolk granules. Okay, this was sort of what Cheyenne did, and then Nikhil joined a few years later. And what he was asking is, really, when you go back, if I can do so, does it really matter that the ooplasm is accumulating perfectly up here and that you get a complete segregation? Is that to somehow biologically meaningful, or could you do with a much more irregular shape you have here and some mixture between yolk granules and ooplasm? So what's the meaning of all that, essentially? And to understand that, what he wanted to see is how cleavages, these are the subsequent processes of embryonic development, depend on this prior segregation of ooplasm and yolk granules. Now, this is what cleavages are looking like in zebrafish. These are meroplastic partial cleavages, and you see 2 cells are formed, and then 4, and so on, 8, 16, 32. And essentially, just to put this in as a schematic representation of cleavages, you subdivide this blastodisc in 2 cells and then in multiple cells eventually, which give rise to the first embryonic tissue, which is the blastoderm. And the blastoderm has then the first cells to become specified, which are the mesoderm and endoderm progenitor cells in this red region here. So what he wanted to know is if how cleavages are occurring in zebrafish, and if they have anything to do with what I showed you before, with the formation of the blastodisc, essentially, and this segregation. Now, the first thing he realized, and he was certainly not the first to realize that, and this goes back to a long sequence of prior papers, that in zebrafish these cleavages are highly synchronized initially. And I just run that. You see, we are just looking at the animal pole now, 4 cells, 8 cells, and so on. As you can see here, the nuclei, they are dividing, the karyokinesis is happening here in a very high synchrony. Then eventually, when this is going on and you go to later cleavage cycles, then these initially very synchronous divisions become metasynchronous, with cells beginning to divide at the animal pole and dividing a little later at the margin of the forming tissue. And then after mid-blastid transition and zygotic genome activation, the entire cleavage has become asynchronous, which is sort of a hallmark of the end of the cleavage period. Now, and this is something I mentioned already, that these cleavages become patterned, and you essentially arrive at a mitotic wave where cells begin to divide at the animal pole, and then they're dividing slightly later at the margin. And this gives rise to a mitotic wave, and what you can then do if you look at a mitotic wave, you can look at the mitotic wave velocity to understand more how that is actually arising. And what is happening is that this delay becomes more pronounced and accumulates over subsequent division rounds, indicating that there is a delay which is getting amplified at each additional round of divisions. If you know something about waves, there are essentially 2 types of waves one can distinguish. One is a trigger wave, which is a biochemical wave, which runs through a common cytoplasm. One should note here that these cells are not completely separated via the cytoplasm. These are partial cleavages. So it could, in principle, be a trigger wave. The alternative, it's a phase wave where every cell is doing whatever it wants to do. It's not communicating, it's not synchronizing, but there might be an overlaying pattern of at factor X, which determines that cells do divide a little faster at the animal pole than at the margin, for example. And these things one need to distinguish to understand how these cleavages are being regulated. And the way he did it is Nickel, he injected histone 1A into one blastomere at the 64-cell stage. This leads to a slowing down of the cell cycle in this one cell in the progeny of the cell. If it would be a trigger wave, this would be overridden very, very quickly by synchronization, by chemical synchronization, and these cells would fall into the pattern of their surrounding dividing cells. If it's a phase wave, these cells would do whatever they want to do, and they don't care really. And the latter one, is exactly what is happening. If you look at that, just look at the blue cells. They divide slow, and they continue dividing slow, and the red cells do their thing. They form this wave, but they don't manage to resynchronize the blue cells, indicating that there's probably very little of a trigger wave, and that we're looking at a phase wave here. So this is just showing you in a schematic, again, that these are the transplanted cells, and then you can show, if you look at the position of these cells along the animal-vegetal axis, and these would be the ones which have received the histone, that they have a longer cell cycle. This is time here. And then this is not being healed after subsequent rounds, indicating that there's no synchronization of these injected cells. Now the question then he asked is, what makes these cells dividing slower at the margin compared to the animal pole? So you looked at S phase and M phase. This is the only phases which are happening during these early cleavages. And it seems to be an increasing delay in S phase and not in M phase. So M phase is staying constant, but S phase is changing. And if you look then at the S phase duration, for example, in different cell rows from the animal pole down to the margin, you see that S phase becomes increasingly longer in these cells. You can then calculate what that, if you just factor that in and S phase delay, how would that theoretically look like? This would be the blue line here, and then the experimental data are the red points, and that falls very much with the expectation that just assuming that S phase lengthens and these delays are accumulating over time, then you would arrive at this mitotic wave you're looking at here. Now, the question then he asks is, how would you get, if you have a mitotic wave, you have to ask where the mitotic wave is really coming from. And, what he then looked at is, the nucleocytoplasmic ratio, which has been implicated already in controlling S-phase lengths in many studies. So we are by far not the first to propose that. And what he sort of speculated then, perhaps it is a change, or, that cells, that the nucleocytoplasmic ratio is different at the animal pole compared to the margin. And this could be the reason for the lengthening of the S phase and the emergence of the mitotic wave he's observing. Now, so the idea is that the nucleocytoplasmic ratio, when it is low, then check1 remains largely inactive. And if you have a high nucleocytoplasmic ratio, meaning that the cells are bigger, and the nucleus remains the same size, then check1 slows down the cell cycle. So presumably this would translate into the idea that cells have a preference to be a little bigger at the animal pole than at the margin, and this could then explain where the S-phase lengthening is coming from. All speculation at this point, and what he had to come up is a couple of good experiments to challenge this hypothesis. And the first thing he did is he looked at the size of the cells, because that was not really known. And he did so at the 8-cell stage, 16 and 128 cell. This is difficult to do cell volumes, 3D rendering, and get really statistically significant data or meaningful data. But what he found really consistently then is that cells at the animal pole, or the future animal pole of this forming blastoderm, have a tendency to be larger than the cells at the margin. So that is sort of consistent with the idea that it could, in principle, be a gradient of cell size translating into a gradient of S-phase lengths and lose the mitotic wave. Now, how can you test that really? Because that's all a correlation up to now, and he did a couple of experiments. I'll show you two, I think. So one is a very simple experiment. He just goes at the 1-cell stage with a pipette in and sucks out a little bit of cytoplasm. In this case, he changes the nuclear to cytoplasmic ratio. What you would then expect is that once they are starting to divide, they will become earlier metasynchronous and eventually asynchronous, because their nuclear cytoplasmic ratio is lower to start with, right? And you can compare that to a control embryo. Control would be here, and this is the asynchrony essentially of the cell cycle lengths here. And you see that the ones which start with a lower nuclear cytoplasmic ratio, a higher nuclear cytoplasmic ratio, are the ones which are becoming earlier metasynchronous. Okay, he did another experiment, and this is an experiment we just repeated essentially, which had been published 25, 30, 40 years ago now, by Chuck Kimball and Don Kane. And it's a very cool experiment, a very, very difficult experiment. And what they did is they removed one nucleus at the 2-cell stage. We're looking right at the animal pole. Then you have a nucleated half of the embryo and an anucleated half of the embryo. And then what is happening is that these guys are dividing here. So their nuclear cytoplasmic ratio is changing over time. And then eventually what is happening by chance is that some of these nuclei are getting pushed out from these cells, and they're ending up now on this large volume of cytoplasm. And the question then he asks is, what would happen to the cell cycle of these cells, because they have a different nuclear cytoplasmic ratio than these cells here. And would they become different by simply being exposed to a different volume of cytoplasm, cytoplasm, yeah. And this is the experiment. Hopefully you can see a little bit of it. These are dividing up there. You're in this sequence here. And then a few are being pushed out. And now you can see that they become asynchronous already, while these nuclei are remaining synchronous because they have a different nuclear cytoplasmic ratio. So they are earlier now in their sort of progression into metasynchrony. And that's what the quantification of this experiment shows. Once you're changing the nuclear cytoplasmic ratio abruptly, you change the division timing of these cells. Okay, so he did then one more experiment, and that goes more in the direction of asking now if cell volume has an influence on the mitotic wave. And what he did is he took the 2-cell stage embryo, he inserted a little object between the two first cells, and then what is happening if you do so, you develop an embryo which has two lobes here, which look like a bilobed embryo, like it would have two blastoderms sitting on the York cell. It's not two blastoderms, but what you have is now you have two gradients of cell volumes. You have cells being larger here, cells being larger here, and then they're smaller here, but also in the valley between the two lobes. And then the question is, how would these, would that lead to two different mitotic waves now, because you have two gradients of cell volumes? Again, it's a very tricky experiment. It sounds easy to do, but it's tricky to quantify. And what he really ends up is indeed with two waves then of two mitotic waves, which both originate at their respective peaks of their lobes. And this is just showing you the mitotic wave and the nearest neighbor distance here. Okay, so what he concluded from this part of the work is that there is apparently, and that was not known, there's a gradient of cell volumes within the developing embryo, and this translates into a gradient of cell cycle length. And the question then is, is that meaningful? Do we have to care about a mitotic wave? Why, perhaps it doesn't matter really too much. So, but then he looked back into literature, and there is quite a bit of evidence that once you have that the length of the cell cycle has an influence on transcription, zygotic gene transcription. And it has been speculated that, if you have, for example, slow cycling cells, they have lesser transcription inhibition and by the replication machinery, so these cells would be the ones which have a higher chance to sort of enter into the onset of zygotic gene transcription than the cells which are fast cycling up here. And so what he thought is perhaps this wave could be important for the onset of zygotic genome activation within zebrafish X. So what he speculated is there's a gradient of cell size translating into a gradient of cell cycle length, and this could be translating into a gradient of zygotic genome activation, where you activate zygotic gene transcription in the cells which are cycling a little slower. And this might have a consequence then for fate specification. I mentioned before that these cells are the ones which are becoming mesoderm and endoderm. Now, this again was pure speculation. The first thing you had to show that, or you had to test if zygotic genome activation is actually patterned within the embryo, which was not known at this point. So he looked here at MIR 430 as a marker for the onset of zygotic gene transcription in zebrafish embryos that had been used before in Xenopus. And this movie is unfortunately a little long, but you will eventually see after a while that zygotic genome zygotic gene transcription is being activated here in the margin. Now you can see the first ones, these bright dots coming up now. It takes time. Now you see a little bit. Here they come. And then this onset of zygotic gene transcription then travels up to the animal pole. So you have an initiation of zygotic gene transcription at the margin, which propagates then to the animal pole. That's very nice, but it doesn't tell you that it has any sort of developmental consequence, really. I just put here a few of these quantifications just to show you that this is not something you see in this movie only. If you look at the animal pole, there's no zygotic gene transcription. If you look at the margin, you already have zygotic gene transcription. You can also do that in these bilobed embryos where you have two gradients of cell volumes and two mitotic waves. And indeed, within the valley, you have the onset of zygotic gene transcription earlier than you have it in the peaks of these two lobes you're generating here. Now, the last question you really had to ask, and that's something which also came with the referees, of course, they were wondering if, it's all nice, zygotic gene transcription, but, what do we learn from it really if it comes to embryo patterning and morphogenesis? So what he did then is he looked at the onset of mesoderm and endoderm cell-fate specification, and he was asking in these bilobed embryos specifically, since they have ectopically earlier zygotic gene transcription in this valley region here between these two lobes, would mesoderm and endoderm being ectopically specified in such a region? And that's exactly what he found then. If you look at CBOX as a readout of mesoderm and endoderm fate specification and a control, it's only in the margin, so only in these marginal cells. And if you look at a bilobed embryo, it's a little hard to see here, but you see that in the valley you get ectopic expression of CBOX, indicating that you get ectopic mesoderm and endoderm specification, which is certainly a meaningful readout for development. So what we now conclude on all that is that there's a cell volume gradient. This goes into a S-phase length gradient, and via the S-phase length gradient or directly via the volume gradient, you can pattern zygotic genome activation and cell-fate specification. One last question was left. And the question then really is, how do you arrive at this gradient of volumes? They don't come for free, so there must be some reason why cells are bigger at the animal pole than at the margin. So what we thought is we want to take the simplest model here and assume that there's a shape of the blastodisc which is given by O-plasmic cytoplasmic streaming you see early on, and from this shape can we derive, can we predict how the division axis is going to be positioned and what would be the resulting volumes of the daughter cells. And so what we did here is essentially showing you on the left side. We start with the first cell. This is after the first division. There's another cell here. And then we are assuming the only thing, and this is very simple, no sophisticated model, we are assuming that the mitotic spindle finds the longest axis of the cell. And if you go back to many studies, Nicola Mink and others, which have shown that this Hertwig rule is a good predictor where the mitotic spindle will go. So we are just assuming the mitotic spindle finds the longest axis, and then we are dividing it halfway, and then we are looking what these volumes would look like. And if you do that simply on cell shape assumptions, not really looking at the real divisions, this is what you would get then here. You would get cells at the animal pole being slightly larger. 1.2 is the ratio then animal pole to the margin in these predictions, and in the observations you see that as well. So indicating that simply assuming that the blastodisc shape determines how the mitotic axis, how the mitotic spindle finds its position, and then how the cleavage position is being defined, you're arriving at these volume gradients. There's one experiment he did then, because referees want to have more, and so what he did is he wanted to change the shape of the blastodisc initially to see if that has a consequence then on the distribution of cell volumes and those on the mitotic wave. And the way he did it, he cut off a little bit of York, portion of the York. Some of these X are healing here, and then you're ending up with something like that, which has a clear higher curvature at this region here. If you then look at the divisions, you get a higher asymmetric distribution of volumes, and this should in principle then derive into a higher exit into asynchrony. So if you're looking at the division timing, these York chopped X here or embryos become earlier asynchronous than the non-chopped ones. This is the real data. And so what we conclude on this work is that you have a geometry of the blastodisk after the first divisions. This, you have these asymmetric cleavages. They lead to a volume gradient, and this volume gradient translates into a mitotic wave gradient, and the mitotic wave gradient then translates into zygotic gene pattern zygotic genome activation and mesoderm and enderm fate specification. Okay, this is a little dark now, but you have to believe me, this is nickel here. Thank you.
Thank you for the great talk. We have time for a couple of questions.
Thank you. Lovely talk, Carl-Philippe. My question is about, as you know, there are axis determinants sitting at the vegetal pole, and then they will be moving up to one side or the other, and then imparting dorsal fate on the side that they end up. So do you see that influence in your cell division rates or anything like that, the cells that become specified as dorsal? And also when you did the removal of the vegetal part of the yolk, did that change anything with respect to any asymmetries there?
So we have not seen any asymmetries along the, presumably, the predicted dorsal-ventral axis. You don't see it before, the first beta-catenin, I think you see first in the dorsal side. So we don't see any clear changes in cell volume or mitotic wave or zygotic genome activation depending on the position along the dorsal-ventral axis. The chopping experiment is a good point, and I think he chops them after most of it has been already transported up to the future dorsal side, because we don't see dorsal-ventral patterning problems. But if you do it earlier, then you would have a problem for sure, yeah.
Yeah, I have two questions, and one is whether you can speculate why there is this S-phase delay in the smaller cells. So could this have to do with maybe making histones during the replicative cycle or something like this, that there might not be enough machinery there in the small cells? And the other one, I was wondering regarding this transcriptional onset, which I think is really interesting, that it happens around the margin where the cells are smaller. So is it sufficient to basically get smaller cells or slow down the cell cycle at the animal pole to get an earlier transcription onset there? Or are there other factors that are required already that are pre-patterning the margin, for example?
So I mean, the first question you answered yourself already. I think it's likely to be histones. From the experiment we did where we're expressing ectopic histones, this indicates that it can slow down the cell cycle and the amount. But we have not systematically looked at the amount of histone. But presumably, since the nucleus size is not changing and the cell size is different, that this translates into some difference in relative amount of histone you have per cell. If that is the case, we don't know, but I think histone, if you ask Nadine Wastenhoven, I think that's probably the hot candidate here. The other question was about... The sufficiency. Okay. If you do the bilobed embryos, I think that's probably the closest experiment where you essentially slow down the cell cycle in the middle where it should be actually longest. There you get early on.
If you do your bilobed experiment, you would have your middle part is very close to the yolk. So I'm wondering whether...
to the yolk, yes, but there's no BISL. I think that's, there's no inducing signal from nodal coming the BISL is mainly sort of on a band surrounding it, but there's not so much BISL at the animal pole. But I see where you're going to, you think about nodal being secreted and using it.
For a last short question.
So I have a question actually. Do you have a feedback on the cell cycle and cell size? So the cell that would divide quicker, they would be maybe smaller because they will be more packed in this area? Or do you see this positive feedback, or don't you have it at all?
So you're thinking about packing of cells and compression or something. If that would affect the volume, I'm not sure, but the density it could change. But so we are not speculating about volume, but we are not speculating about cell density. We are speculating only about the nucleus-cytoplasmic ratio. That's, since these are reductive cell divisions, there's always halving in each division.
Squeezing a very last one.
So what is your model of, so the fact that the ZGA turns on precociously would influence cell fate? You think so
Yeah.
But then,
very good question.
yeah.
You know, why would they become mesoderm and endoderm? And again, this is pure speculation. One could argue that they need to express receptors for nodal, being competent to respond to nodal signaling and being, you know, able to essentially translate that into mesoderm and endoderm cell fate specific gene transcription, which would be a CBOX, and these are, you know, typically coinciding with zygotic genome activation. Yeah.
All right,
Okay.
let's thank our Philippe again. And our next speaker is going to be Marie-Emilie Terret from Collège de France, and she's going to speak about biophysics of oocytes divisions.
So I actually need the things to pass the slides. So it's this one? Thank you. Okay. So first, I would like to thank Marie-Hélène and Elvan for organizing this wonderful meeting and for inviting me, and also the Fondation Singer-Polignac for the venue, which is quite exceptional. So today I'm going to talk about biophysics of oocyte division, but also a lot about mechanical properties and oocyte quality. Okay. So just to give you some numbers, so infertility, and we heard a lot about it this morning, is a major problem worldwide. So here are some numbers in France. So my pointer doesn't work anymore. No. Does someone has a pointer? So this one maybe works. Yeah, okay. So here are some, it's a major problem worldwide. So these are some numbers in France from 2021 and the last perinatal survey. So it was estimated that 10% of couples were affected by infertility, that 3.6% of babies were born through assisted reproductive technology or ART, and that with a moderate success rate of only 21%, and a cost of 300 million euros for social security. So the success rate of ART is linked to the quality of the gametes, so the oocyte and the sperm, and it is estimated that in 1/3 of the cases, the problem comes from the mother, in 1/3 of the cases from the father, and in 1/3 of the cases, either both or we don't know. So in the lab, we are interested into oocyte production, and as you heard a lot about this morning, it's an error-prone process. So it generates a high basal rate of poor quality oocyte that increases with maternal age. So this is actually the same graph as the one you saw this morning, with the percentage of trisomy in regard with the maternal age. So you have this curve, so it would be a bell curve, but it's cut a little bit short. So it increases with maternal age, and concomitantly, you have an increase of the mean age of the mothers at birth of first child, which in France was 31 years old in 2021. So it brings the question of what defines oocyte quality and how do we recognize high-quality oocytes. Something striking at fertilization, and we heard about that yesterday also, is the size difference between the oocyte here and the sperm that you can see here enlarged. So at fertilization, to produce an embryo, the sperm will bring mostly its genome, but in the oocyte, you will have the genome from the oocyte, but also all the cytoplasm. So right away, you see that oocyte quality is not all about DNA, but it's DNA and also cytoplasmic content that will be passed on the embryo that has roughly the same size as the oocyte before fertilization. So oocytes are produced before birth, as you heard also about during this congress. They will stay quiescent until puberty, and cyclically after puberty, some will undergo a growth phase where they will accumulate mRNA and proteins in their cytoplasm, which correspond to an active phase of transcription. So then oocytes are going to enter into the first meiotic division, they will form a spindle where the nucleus was, which is more or less in the center of the cell. Then this spindle will migrate to the cortex of the cell along its long axis, leading to a very asymmetric division in size, producing a big oocyte and a small cell, the polar body. And the asymmetry in size of the division is very important to keep all the maternal stores accumulating during the growth of the oocyte in the cytoplasm to be transmitted to the embryo. So then at fertilization here, fertilization will trigger a second meiotic division, which will be also asymmetric in size because the spindle in oocyte arrested in metaphase 2 is already under the cortex. And then it will create an embryo here, a zygote, that has roughly the same size as the oocyte before it divided. So these two asymmetric divisions in size really ensure to minimize the loss of volume of cytoplasm in the polar bodies to pass the maximum of volume to the embryo at fertilization. And so oocytes must have a correct ploidy, but also sufficient maternal reserves to produce healthy embryos. So in the lab, we have shown in the past that cortical tension, and we and others, that cortical tension is a key regulator of oocyte development. So cortical tension is a mechanical property that regulates cell stiffness, and it is in part regulated by cortical actomyosin spatiotemporal organization. So this is an oocyte in prophase before entering into the division with this big nucleus here and the DNA in blue. And it will accumulate myosin 2, which is a protein that can bind to actin networks and exert forces. And so these oocytes are stiff. Then when they enter into first meiotic division, they will nucleate an actin layer under the cortex, as you can see here, progressively. And this will chase myosin 2 from the cortex, decreasing cortical tension and making these oocytes softer. So these are soft. And so that myosin 2 that was cortical will go in the cytoplasm and on the spindle, and it will participate in spindle positioning from the center to the oocyte cortex, leading again to this asymmetric division in size, which is important to keep the maximum cytoplasm in the oocyte. Interestingly, variation in cortical tension exists naturally. We can measure them with biophysical techniques such as macropipette aspiration and atomic force microscopy, and you see that you have a variation in cortical tension in prophase oocyte, with some oocytes being more stiff than others. And this reflects actually the variability in cortical actomyosin organization. Here, for example, you see myosin 2 cortical enrichment, and some oocytes have a lot of myosin 2 at their cortex and some have less. It is known also that cortical tension defects exist naturally and can affect oocyte development. And we have shown, for example, that oocytes that are too soft have difficulties to capture their chromosome, to put them on the metaphase plate, leading to a defect in segregation at anaphase and production of oocytes that are aneuploid in meiosis 2. So today, I will talk about something that is not published, so the consequences of excessive cortical tension on oocyte development, and it's a project that is undertaken by Anastasia Chiabi, in the audience, a PhD student in the lab. So to be able to assess the consequences of excessive cortical tension, we wanted to increase the number of oocytes that we could study, and for that we sought to engineer stiff oocytes. So to engineer stiff oocyte, we use a tool to force active myosin 2 at the cortex. So this tool was developed by the Rongley lab, where they fused a ROAGF with a cortical anchor. So it localizes this ROAGF at the cortex, and it will activate the ROA-ROC pathway to activate and recruit active myosin 2 to the cortex. And this is what you can see here. So there are two oocytes in meiosis 2, one control and one expressing this probe. And you see in blue that active myosin 2 is indeed recruited at the cortex. And these are meiosis 2 arrested oocytes. So in our case, we modified a bit this tool and we fused this ROAGF to esrin, which is a protein that is always at the cortex at all times during the division. And we actually express this construct in prophase oocyte to increase cortical tension starting from meiosis on. When we did that, we verified, of course, by immunofluorescence that we were able to recruit myosin 2 to the cortex, which is the case, and that was sufficient to increase cortical tension, as you can see here. So these oocytes are the ones that express this construct at the cortex and that will recruit myosin 2 to the cortex, and I will call them stiff for the rest of the talk. And the control oocytes are either oocytes that are non-injected or that are only injected with esrin, which is again the cortical anchor that is there to put myosin 2 to the cortex. So we have this increase in cortical tension, and we wanted to see the effect on oocyte divisions. So this is a control oocyte. So oocytes are still in prophase, so the nucleus would be here, and you can barely see it. So the nuclear envelope breakdown will occur. A spindle will form during these 4 hours after nuclear envelope breakdown. Then this spindle will migrate from this period of time, leading to this asymmetric division in size at anaphase. So in stiff oocyte, everything occurs normally. So the division timing is comparable to controls, the division geometry as well, with asymmetric division in size, and the division efficiency is also comparable to control. But however, if you look at the cortex here, it seems a bit different to that of the control oocyte. And so we wanted to gain more insight into what was happening at the cortex. So for that, we followed, so here are control oocyte expressing the cortical anchor, and you see that this cortical anchor is localized homogeneously at the cortex until anaphase, as you can see here, and that oocytes are not polarized, they do not show any sign of polarization before the chromosome have moved to the cortex. So in stiff oocyte, on the contrary, you start seeing some patches of accumulation of the probe, the ROAGF, which again is a readout for myosin 2, so here. And these patches are progressively coalescing into one big region here of polarization that is always away from where the division will occur, as you can see here. And this is a very penetrant phenotype because all oocytes expressing this probe are going to be stiff and will divide and will polarize that way. So again, to gain even a higher magnification of what is going on at the cortex, in collaboration with the lab of Hervé Turlier, we reuse the plugin that they developed for this paper, where they can actually follow cortical flows by following the speed of movement of fluorescence in the cortex. So in this graph, what you can see here is from NEBD to NEBD plus 8 hours, so at the entry of meiosis 1 up to the exit of meiosis 1. So here you have flattened cortices of oocyte with 180 degree, that would be the polarization site, and 0 degree and 360 where the division would occur. So you take the oocyte and you just unfold it so that it's flat. And here, the colors, the brightest, the highest the speed. And so what you can see at the beginning of the division is that not much is happening. So you have some movement of fluorescence in the cytoplasm, but that are very low in intensity. And progressively, they are going to merge in these zones that will form this polarized region here that will be opposite to where the division will occur. So we can measure the speed of this polarization, and so it's pretty fast up to beginning of meiosis until mid-meiosis 1, and then it plateaus once this polarization site is organized and is maintained. So it has been shown in other cells, and in particular by the lab of Carl-Philippe, that if you increase cortical tension, you will polarize your cells spontaneously, and that it will change the mode of migration of cells. So oocytes are not migrating cells, so we wondered if this polarization, instead of moving the cell, could move the inside of the cell. And so for that, we looked at flows that were occurring in the cytoplasm of these oocytes. So on the left, so these are sticks maps from NEBD plus 4 hours to NEBD plus 8 hours, so when the spindle is migrating. In control oocytes, so you don't see much here, but you have some flows, but that are not directional. You don't see any directionality, and they are very faint because, again, you have vectors here that represent the directionality of the flow, and their intensity is color-coded so that the whiter, the strongest intensity. So here you have a lot of arrows that are not directional and that are pretty purple, meaning that they are not strong. In stiff oocytes, so this would be the polarized site, and here where the division will occur. So first you have flows that are along the cortex, which are reminiscent of the cortical flows that I just showed you before. But you also have strong flows that originate from the polarized region and that go away from it here where the division will occur. And in fact, these flows are higher in stiff oocytes compared to controls because you can plot the velocity of these vectors and you see that it is higher in stiff oocytes compared to controls. So flows are originating from this polarized region. They are very directional, they are very strong, so can they move objects in the cytoplasm? And so for that, we injected the first inert objects, such as oil droplets, in oocytes. So in the past, we have shown that if we inject these oil droplets, so here in white, they are going to stay at the center of the cell during the division, near the geometrical center of the oocyte, here in yellow. And so in stiff oocyte, we did the same. We injected oil droplets, and we could show that these oil droplets were actually pushed away from this polarized zone in the direction of division. And so we measured the displacement of these droplets, and it took them 3 hours and 20 minutes to be displaced. Then they stop moving, and then you have anaphase. So, okay, so we create flows from the polarized region of the oocyte. They can push inert objects, but can they push more physiological objects that are big enough to resemble hot droplets, such as spindles? So this is a control oocyte and a stiff oocyte. So in this control oocyte here, it's at an EBD plus 2 hours. So the spindle forms in the center of the cell, the oocyte, and you will see that it will progressively migrate along its long axis toward the closest cortex. So this is what you can see here. So the spindle is forming, it will move there, and then you will have anaphase with the asymmetric division in size. So in stiff oocyte, the spindle also forms in the center of the cell, but you see that when the polarization zone is created, it moves away from it and not in the same orientation. So we measured the timing of spindle positioning in control and stiff oocyte. So in control, it takes 4 hours, and in stiff oocyte, it takes 3 hours to displace, which is more comparable to the hot droplet displacement. So then we looked also at the difference in angle between the axis of the spindle and the axis of migration. So in control oocyte, these two axes are roughly superposed, so the angle would be very small, and this is the case here. Whereas in stiff oocyte, sometimes the spindle migrates perpendicular to the axis of migration, which is shown here. We also computed another metric, which is the difference between the axis of the spindle at the beginning and at the end of migration. So in controls, again, this angle is very low because the spindle migrates in a very directional manner. Whereas in stiff oocyte, you see here that it's much more dispersed. And so some spindles are rotating actually a bit when they are migrating. And we are again collaborating with the team of Hervé Turlier to understand the migration of these spindles depending on their position at the beginning of migration. So the flows are able to push objects, they're able to push the spindle, but can they move also more discrete objects in the cytoplasm, such as organelles? So we looked at mitochondria first. So these are control oocytes 1 hour after nuclear envelope breakdown. The mitochondria are distributed throughout the cytoplasm, and this would be the hole where the spindle will form and the chromosome are. And so you see that the division proceeds, and the mitochondria are going to gather around this spindle and be depleted a bit from the rest of the cytoplasm. So in stiff oocyte, mitochondria are also distributed through the cytoplasm, but then they are going to be pushed away from the polarized region, and they will be mainly localized in the other hemisphere of the oocyte. So this is in meiosis 1, and we wondered what could be the effect of having this pushing of the organelles in meiosis 2 oocyte. So could these organelles be pushed, for example, in the polar body and be extruded and lost in meiosis 2 arrested oocyte? So first, we quantified the number of the amount of mitochondria in control and in stiff oocyte, and indeed, there are less mitochondria in stiff oocyte arrested in metaphase 2 compared to control oocyte. We also measured other organelles, such as Golgi, as you can see here, and you also have the same trend, with less Golgi being present in metaphase 2 arrested oocyte in stiff oocyte compared to control oocyte, suggesting again that this pushing of the organelles in meiosis 1 is sufficient, even if you have an asymmetric division in size, to deplete these organelles from the stiff oocyte. So these oocytes are ready to be fertilized. So we looked at some important players for fertilization that you heard about yesterday. So we looked at CD9, Juno, and microvilli. So these are control oocytes. So at the beginning of the division in meiosis 1, these players are localized all around the cortex of the oocyte. Then they progressively become polarized as the spindle migrates to the cortex. So it will create a differentiated zone where Juno, CD9, and macrovilli will be not present, and they will cover the rest of the oocyte. And it's the same in meiosis 2. The macrovilli, CD9, and Juno will be opposite to where the DNA is. And it's important because at fertilization will trigger the second meiotic division, so you don't want your parental genomes to fuse too early. So in a way, making sure that you will be fertilized away from this DNA safeguard for this precocious mixing of the genomes. So in stiff oocyte, all these CD9, Juno, and macrovilli are going to be, in a way, localized in patches early on during meiosis 1. Then they will polarize strongly in this zone that is away from the division. And it's the same in meiosis 2. they will form a highly polarized region that is much restricted if you compare to control oocytes. So the zone where the sperm can bind is restricted in this stiff oocyte compared to control oocytes. And so we looked also at cortical granules, which are important to avoid polyspermy. So these granules, when the oocytes are fertilized, are going to be released from the oocyte, and they will prevent other sperm to bind to the oocyte. So in control oocyte, here, you see that the cortical granules are going to be translocated to the cortex of the oocyte, and they will follow the same pattern as you just saw before with sedenine and Juno. They will be actually extruded from the zone where the spindle is, and they will accumulate here. So in stiff oocyte, they will polarize precociously in mid-meiosis 1, and this polarization will stay until meiosis 2, where they will be strongly accumulated in a very small area zone of the oocyte. So to conclude this part, I showed you that stiff oocyte in prophase 1, because of the accumulation of myosin 2, which is something that we see naturally in oocyte, is going to create a polarization mid-meiosis 1 with increased and directional cytoplasmic flows emerging from this polarized region. So it will induce altered spindle migration, with the spindle being able to be pushed by these flows in various directions. And it will also create altered organelle distribution and amount, especially mitochondria and Golgi, but also aberrant localization of cortical granules and CD9 and microvilli. So we are now looking at the effect for fertilization. Do we have a suboptimal fertilization because of this restriction of where the sperm can bind? And do we have polyspermy also due to this, again, restriction of cortical granules? And we also would like to look at embryonic development to see if it's impaired, especially because of this depletion of organelles in the oocyte in meiosis 2. So of course the question we have, so everything I showed you up to now was in engineered stiff oocyte. So the question is, does this phenotype exist naturally in naturally stiff oocyte? So for that, in collaboration with Gaëlle Le Thor, we are using oocytor, an AI-based tool she created that we can use to compare oocytes and extract the most striking features between oocyte population. So we compared the control and stiff engineered mouse oocyte. We extracted a stiff score that is the most discriminating features used to discriminate this population. We applied these stiff scores to about 300 movies of maturating mouse oocyte, and we could see that 10% of these natural mouse oocyte display the same phenotype as in engineered mouse oocyte, suggesting that this phenotype does exist naturally. So we want now to check if some human oocytes naturally present the same phenotype as stiff mouse oocytes. And for that, in collaboration with Xavier Paulet-Villard and Mohamed Yahya-Tien, we want to apply these stiff scores to human oocytes. Indeed, they have a database of maturating human oocytes, about, again, 300 movies that we used in other projects that we want to run our stiff score on to see if we could see some of the phenotypes. And this is one example of an oocyte that could display this phenotype with a flattening here in the region that could polarize and a change in the aspect ratio of these cells that we also see in the mouse. So just to finish, I told you that cortical tension, but more generally, mechanical properties are important to regulate oocyte quality. And so now we would like to use these mechanical properties as biomarkers of oocyte quality. So very briefly, Lucie Barbier, a postdoctoral fellow in the lab, so she designed a macroscopic device for oocyte mechanical screening. So it's a very simple one. It's a constriction-based macroscopic device where you can push oocyte with a controlled flow. This constriction is smaller than the diameter of the oocyte, so you can record oocyte deformation and entry into the constriction, the timing of entry, timing of passage, and so on. She could show that this method identifies oocytes with altered mechanical properties by comparing control and soft engineered oocytes. And she could show that it is non-invasive because if you pass zygotes in this chip, they are going to develop normally up to the blastocyst stage. So of course, we would need more experiment to show that it is non-invasive, but we would like also to scale up this device for human oocyte. And before doing that, we had actually to assess the mechanical properties of mammalian oocyte. And so this is the project of Rose Bulto, a PhD student in the lab, and she will talk actually in the next session, so I will be very quick on that. But she characterized using this technique elasticity, viscosity, and cortical tension of human and mouse oocyte. She could design a new model to extract these mechanical properties from this force curve that you obtain with AFM. And just showing cortical tension, but she will show you much more. She showed in particular that cortical tension of mouse and human oocytes differ in meiosis 1. So they decrease in the mouse and they stay the same value in human. But you have this same difference between prophase and meiosis 2, and they are in the same range, suggesting that the kind of tool that we design here could be used also in human oocyte, and this is what they will test next. So with that, I would like to thank all the people that participated in this project, and I already mentioned them during the talks, very good, talented and fierce PhD and postdocs. So in blue, this is the Noemi talked yesterday, and Rose will talk in the next session again. All our collaborators, the facilities where we did the AFM in Paris and in Lyon. And also, we have a 3 years postdoctoral position available, so if you're interested, please contact me. Thank you for your attention.
Thank you, Marie-Emilie. We have time for questions.
Hi. Yes, thank you for the talk. There is one thing that I don't understand. So you're saying that by overexpressing your actin polymerizate on the cortex, so you have more actin and that's stiffer.
More myosin 2. Yeah.
More myosin 2, sorry. Yeah. More myosin 2 that's stiffer. But then when they resume meiosis, all of this relocalize to the opposite part of the exactly. Polar body extrusion. So does the other part of the oocyte, is it still stiff?
No, we don't think so, actually. So it's stiffer at the beginning, but then it creates an imbalance and it's going to break symmetry. But then it's only at the beginning in prophase, and then once you enter into the division, since you have this polarized region that will merge, then they are not stiffer anymore. So in this so we need to verify that. But in the region where they have the polarization, of course, I think that it will be stiff, and the other region it will not.
So would it be even softer than the control oocyte?
So that may be, because you would deplete in a way maybe myosin 2. So yeah, this is something that we can measure, but we did not yet.
Okay, thank you.
Just a quick question. I was curious if you've looked at oocytes from older mice and if they have different stiffness parameters.
So we looked at them, so I'm not going to tell a lot about it because Rose is going to say more. But yes, indeed, their mechanical property decreases. So we are not the only one who showed that. I mean, other labs also, but they are decreasing with age and they are become softer. And so softer would mean maybe with poorer chromosome segregation, as we showed before, but also maybe more receptive to forces coming from the environment of the ovary in a way. So especially if you think about fibrosis, with the ovary being stiffer, I mean, it could also transmit more the forces to oocytes that are softer than normal oocytes, let's say.
That's very, very nice talk. Thank you. Where the organelles disappear? I mean, I didn't. So, the mitochondria. So, I mean.
So that we have to check.
It's the polar bodies, or
yeah. So this is what we think, but we have to prove that. We didn't prove it yet. So the polar body. So we didn't quantify, but by eye they seem to have the same size for sure. So asymmetry in size. And so what we think is that these organelles are pushed. And when they divide, some are going to be pushed in the polar body, and that's why you get depletion in meiosis 2, even if you still have this asymmetric division in size. So it's difficult to follow. So we have some movies where we see that, but the problem is that, I mean, with the drugs to see the organelle, they get exhausted at one point, so it's difficult to see. But again, this is the hypothesis we have, that in a way, you divide asymmetrically, but it's as if you would divide symmetrically because you push the organelles in the polar body.
Asmin at cross-link membrane and actin, so will overexpress asmin itself will change the stiffness.
So not in this system. So this is work that we did in the past and that we actually showed here, is that if you overexpress actin in mouse oocyte, it doesn't do anything. So it doesn't increase cortical tension, it doesn't change any parameter that we measured in terms of viscosity, elasticity. So it seems to be, so either you have already a lot and putting more doesn't matter, but it doesn't change the mechanical properties of the cell, nor the division timing, nor anything. Yeah.
If not, let's thank Marie-Emilie again. going to have a coffee break, which is according to program until 4 p.m., but maybe we can gather 15 minutes before so that we have a little bit less time pressure for the short talks, if works for the program. So we'll gather again at 3:45. Okay, sorry, small change. We are gathering at 3:30 so that we are going to finish a little bit earlier and having more time for the short talks.